Picture a newborn baby. Doesn't matter where in the world. London, Lagos, Seoul, Sao Paulo. That infant arrives with skin that hasn't yet fully developed its pigmentation. For the first few weeks of life, the color isn't quite settled. There's a biological reason for that delay, and it hints at something extraordinary. Skin color in humans is not a fixed feature baked into existence from the beginning of time. It is a moving target, a negotiation, a response. And the story of how it became what it is today is one of the most surprising, most layered, most thoroughly misunderstood chapters in the entire history of our species. Because the truth is, if you went back far enough that no written language existed, no cities, no agriculture, no concept of nationality or race, every single human being on this planet would have looked far more similar than most people today ever imagined. There's a thought experiment that biologists sometimes use. Imagine you could compress 300,000 years of human history into a single hour. In that hour, for the first 56 minutes, every human alive would share essentially the same skin tone. Not similar, the same. A deep, rich brown, dense with the pigment that evolutionary biology had spent hundreds of thousands of years carefully selecting and protecting. It wouldn't be until the final 4 minutes of that compressed hour, a geological eye blink, that the remarkable diversity of human skin tones we see on Earth today would begin to emerge. And the reason it emerged when it did, where it did, and in the specific way it did, is rooted not in culture, not in geography alone, but in something as invisible and indifferent as the ultraviolet radiation streaming from a star 93 million miles away. To understand what happened, we first have to understand skin itself, not as the body's exterior coating, but as one of the most biologically sophisticated organs the human body possesses. Most people have never thought seriously about what skin is doing at any given moment. Right now, as you read this, your skin is regulating your body temperature, sealing out pathogens, detecting pressure and temperature and pain, waterproofing your internal organs, and synthesizing molecules your body cannot obtain any other way. It is simultaneously a barrier, a sensor, a factory, and a shield. And one of its most critical long-term functions, one that shapes the entire trajectory of human physical appearance, is its interaction with ultraviolet radiation. UV light is not visible. You cannot see it or feel it arriving on your skin, but it is affecting you every single moment you spend outdoors. And whether that effect is protective or destructive depends almost entirely on one molecule: melanin. Melanin is the pigment produced by specialized skin cells called melanocytes. It's not just one molecule, it comes in multiple forms, with eumelanin producing darker tones and pheomelanin producing lighter, reddish-yellow ones. But the critical property both share is the ability to absorb ultraviolet radiation and convert it into harmless heat before it can penetrate into the deeper layers of tissue where your cellular machinery lives. Think of melanin as a microscopic radiation shield distributed through every layer of your skin. The more of it you have, the more UV is intercepted at the surface. The less UV that reaches the nucleus of your skin cells, the less chance there is of UV radiation doing what it does best to DNA, breaking chemical bonds, creating mutations, scrambling the molecular code that controls cellular behavior. Those uncorrected mutations, accumulating over years and decades, are the biological foundation of melanoma, the deadliest form of skin cancer. In environments where UV radiation is constant and intense, like the equatorial regions of sub-Saharan Africa where Homo sapiens first appeared, high melanin concentration is not an aesthetic preference. It is a survival mechanism with direct consequences for whether you live long enough to have children. When paleoanthropologists and geneticists trace the early emergence of Homo sapiens, they converge on a remarkable picture. Between 250,000 and 350,000 years ago, the earliest anatomically modern humans were living in Africa, and the evidence strongly suggests they were deeply pigmented. This isn't guesswork. Researchers can use comparative genetics to model ancestral skin pigmentation by tracing gene variants across contemporary populations. They can also look at our closest primate relatives. Chimpanzees and gorillas have dark, heavily pigmented skin beneath their fur, which evolved independently but for similar UV protective reasons. The genetic variants that confer high melanin production in humans appear to be ancient, deeply conserved, and widespread across populations with continuous ancestry in equatorial regions. The scientific consensus today is that heavily melanized skin is the ancestral human condition, the biological baseline from which all variation descended. But there's a second reason heavily pigmented skin was so critical in early human Africa, and this one is almost never discussed outside of specialist academic literature. It involves a molecule most people only know from prenatal vitamin supplements: folate. Folate, the naturally occurring form of vitamin B9, is not optional biology. It performs essential functions in the synthesis and repair of DNA, in the regulation of gene expression, and most critically, in the earliest stages of embryonic development. In the first 28 days after conception, often before a woman even knows she is pregnant, the embryo's neural tube, the precursor to the brain and spinal cord, depends entirely on adequate folate to close properly. When it doesn't close, the consequences are catastrophic: spina bifida, anencephaly, miscarriage. Folate deficiency doesn't just make pregnancy harder. In severe cases, it ends human lineages before they begin. Here is the problem. Ultraviolet radiation destroys folate in human blood. UV light penetrates the skin, reaches the circulatory system, and breaks down folate molecules in the bloodstream. This was demonstrated definitively by dermatologist and anthropologist Nina Jablonski and her colleague George Chaplin in landmark research published in the early 2000s. Work that fundamentally reshaped scientific understanding of why skin color varies across human populations. Their insight was that in high UV environments, the body faces a dual threat: direct DNA damage from UV penetration into skin cells and systemic folate destruction that compromises reproduction. Both threats are addressed by the same biological solution: high melanin concentration. More melanin means UV is absorbed before it reaches the bloodstream. Folate is preserved. Pregnancies succeed. More descendants carry the same melanin-producing genes. Repeat for 200,000 years. You end up with an entire species in deep Africa where anything less than substantial melanin concentration is a meaningful reproductive disadvantage. The selection pressure wasn't just about cancer. It was about whether your children were born at all. Now consider what happens when that same species begins to move. Between 60,000 and 80,000 years ago, though some evidence suggests earlier excursions, groups of modern humans began migrating out of Africa in earnest. This was not a planned exodus. There was no map, no destination, no awareness of the scale of what was happening. These were small bands, almost certainly following prey animals, tracking coastlines, moving incrementally through generations. A group shifts its territory slightly north. Their grandchildren move a little further still. Over centuries, then millennia, modern humans spread through the Arabian Peninsula, into South Asia, across Southeast Asia, into Europe, eventually into the extreme north of Eurasia, and ultimately across the Bering Land Bridge into the Americas. The movement was slow, discontinuous, and punctuated by long periods of settlement and genetic bottlenecks. But the environmental shift it entailed was dramatic. At 50 degrees north latitude, roughly the latitude of London or Moscow, the annual UV radiation reaching Earth's surface is approximately a quarter to a third of what strikes the equatorial regions of Africa. The angle of sunlight is lower, the atmosphere is thicker, winters are long and dark, and cloud cover is frequent. The biological implications of this change are not subtle. They are stark, systemic, and ultimately lethal for those unprepared for them. In a high UV environment, melanin is a shield. In a low UV environment, that same melanin becomes, in a very specific and consequential way, a liability. Not because UV damage is no longer a concern, it is still present and still causes cancer, but because in the far north, the UV radiation available is not just a hazard to be blocked, it is also a resource that the body desperately needs. Vitamin D is synthesized in the skin when UVB radiation, a specific wavelength band of ultraviolet light, strikes a cholesterol compound called 7-dehydrocholesterol in the epidermis. The reaction converts that compound into a precursor molecule that the liver and kidneys then process into the active hormone we call vitamin D3. The word vitamin is technically a misnomer here, because true vitamins must be obtained from food. Vitamin D the body can make itself, provided it has sufficient UVB exposure. But in the low UV north, that synthesis becomes extremely limited, especially in winter months when UVB levels drop to near zero across much of Europe and Northern Asia. And vitamin D is not a luxury micronutrient with marginal effects. It is a master regulator of calcium metabolism, immune function, and muscle physiology. Without it, calcium from food passes through the digestive system largely unabsorbed, regardless of dietary intake. The skeleton literally cannot mineralize correctly. The clinical consequences of severe vitamin D deficiency, as documented extensively in pediatric medicine long before anyone understood the mechanism, are devastating. In children, the condition known as rickets causes bones to soften and deform as they grow, producing the characteristic bowed legs, pigeon chest, and thickened wrists that were tragically common in northern populations throughout history. In adult women, deficiency causes a different but equally serious condition: osteomalacia, a softening of already formed bone. And in pre-industrial populations, where women underwent multiple pregnancies without medical support, softened pelvic bones were not merely uncomfortable. They made labor mechanically dangerous in ways that killed both mothers and infants at rates that compound catastrophically over generations. Vitamin D deficiency in a pre-supplementation world at northern latitudes was a slow demographic crisis written in bones. Now place a heavily melanized individual in that environment. Their skin, optimized over hundreds of thousands of years to block UV radiation efficiently, continues doing exactly what it was designed to do. It blocks the UV. All of it, including the UVB wavelengths the body needs to synthesize vitamin D. The melanin cannot distinguish between UV wavelengths that damage DNA and UV wavelengths that initiate the synthesis pathway. It simply absorbs and neutralizes. For someone whose ancestors lived for 50,000 generations in equatorial Africa, the system worked perfectly. For someone now living at 55 degrees north latitude, with short winter days and perpetual cloud cover, that same system is now starving the body of a molecule it cannot survive without. The evolutionary mismatch is not subtle. In a northern winter, a highly melanized individual may produce almost no vitamin D from sun exposure for 3 to 5 months of the year, regardless of how much time they spend outdoors. This is the mechanism that drove depigmentation in northern populations. It was not aesthetic selection, not cultural preference, not any force related to how humans perceived each other socially. It was bone disease, infant mortality, and maternal death in childbirth, recurring generation after generation, selectively reducing the reproductive success of individuals with higher melanin concentrations in environments where UVB was scarce. Mutations that reduced melanin production, that slightly thinned the shield and allowed slightly more UV to pass through, were already present in the population at low frequency, as random mutations always are. In Africa, those mutations offered no advantage. They might have been mildly disadvantageous, slightly increasing cancer risk or slightly reducing folate protection. But in the north, each generation that survived the winter was slightly better represented among those with reduced melanin. Not dramatically, not all at once, but consistently, over thousands of years, in one direction. What makes the timeline of this change so surprising is how recent it is. For a long time, the prevailing assumption in anthropology was that northern European populations had been gradually lightening in skin tone ever since the first modern humans arrived in Europe tens of thousands of years ago. Ancient DNA analysis, made possible only in the last 15 years by dramatic advances in sequencing technology, demolished that assumption with remarkable precision. The ancient genomic data now available from European archaeological sites reveals something that challenges almost everyone's intuitions about human prehistory. The hunter-gatherers who lived in Europe for the first tens of thousands of years after the out-of-Africa migration were not pale. They had significantly darker skin than modern northern Europeans, even after living at northern latitudes for a very long time. The most striking single piece of evidence for this is a 10,000-year-old individual recovered from Cheddar Gorge in southwestern England, known in the scientific literature and popular press as Cheddar Man. When researchers at University College London and the Natural History Museum published his ancient genome analysis in 2018, the results stunned the public. The genetic markers associated with skin pigmentation suggested Cheddar Man almost certainly had dark to medium dark skin. He also, intriguingly, had the genetic variants associated with blue eyes, a combination that strikes modern eyes as paradoxical, primarily because we've come to mentally link very pale skin with blue eyes. In Mesolithic Britain, that association didn't exist yet. These features had not yet co-evolved into the package we recognize today. Cheddar Man is not an anomaly. Multiple ancient European individuals sequenced from this period show similar profiles: dark skin, blue or green eyes, dark hair, a combination that would look genuinely exotic in a modern European context. If hunter-gatherers were still dark-skinned after tens of thousands of years in Europe, what changed? What drove the dramatic shift to the very pale skin typical of northwestern Europeans today, the kind that burns after minutes in a summer sun? The answer appears to involve one of the most consequential transitions in all of human history: the Neolithic Revolution. Beginning around 10,000 to 12,000 years ago in the Fertile Crescent and spreading across Europe over the following several thousand years, the shift from hunter-gatherer lifeways to settled agriculture fundamentally transformed the human diet. And that dietary transformation didn't just change what people ate, it changed the selection pressure on their skin. Hunter-gatherers consumed an enormous variety of foods, and that dietary variety included significant quantities of foods naturally rich in vitamin D. Fatty fish, salmon, mackerel, herring, sardines are among the most concentrated natural sources of vitamin D in existence. Wild game, particularly organ meats and fatty cuts, contributes meaningfully to vitamin D status. Shellfish, sea mammals where available, and eggs all contribute. This dietary portfolio wasn't an accident or a luxury. It was the natural consequence of eating a diverse range of animal products across seasons. Vitamin D from food, absorbed through the gut rather than synthesized in the skin, doesn't require UVB sunlight at all. It bypasses the whole equation. A hunter-gatherer at 55 degrees north latitude with dark skin and a diet rich in fatty fish could maintain adequate vitamin D status through winter without any sun exposure whatsoever. The Neolithic farmers who replaced them and eventually interbred with them ate primarily cereals: wheat, barley, millet, rye, oats. These are almost entirely devoid of vitamin D. Agriculture produced reliable caloric abundance in the form of carbohydrate-heavy grain, but it essentially eliminated the dietary vitamin D that had helped northern hunter-gatherer populations survive dark winters with darker skin. Suddenly, populations that had been managing on a combination of skin-synthesized and diet-sourced vitamin D were now almost entirely dependent on what their skin could produce from weak northern sunlight. And if their skin retained high melanin concentrations, it couldn't produce nearly enough. The nutritional floor dropped out. The selection pressure for lighter skin intensified dramatically, not because lighter skin was a new idea, but because the dietary safety net that had softened the pressure had been removed. This is why the archaeological and genetic evidence shows such rapid depigmentation in European populations during the Neolithic and subsequent millennia. The change that took tens of thousands of years to barely begin during the hunter-gatherer period happened by evolutionary standards in a relative sprint, perhaps 5,000 to 8,000 years. The genetic variants most strongly associated with pale skin in modern Europeans, including a variant in a gene called SLC24A5 that affects melanin transport within skin cells, appear to have been at very low frequency in European populations before the Neolithic, and then spread with dramatic speed as farming populations expanded. This is not gradual drift. This is strong, positive natural selection operating over a compressed time frame. The algorithm, as it were, found the answer it had been looking for and ran with it. The genetics of skin pigmentation are far more complex, however, than a single gene story. Skin color is what geneticists call a polygenic trait. It is the cumulative output of dozens of genes acting in concert, each contributing a small effect to the overall phenotype. At least 6 genes have major documented effects on skin color in humans, and dozens more have smaller modifying influences. These genes affect not just how much melanin is produced, but what type, how it is packaged into granules, how those granules are distributed through the skin cells, and how the skin responds to UV exposure over time. The result is not a binary switch between dark and light, but an extraordinarily fine-grained continuous spectrum with thousands of possible points. What makes the genetics even more remarkable is what happens when you compare populations that independently evolved lighter skin in different parts of the world. East Asian populations have substantially lighter skin than equatorial African populations, as do Northern European populations. The selection pressures they faced were similar: reduced UV at higher latitudes, selection for vitamin D synthesis. But when geneticists examine which specific gene variants are responsible for lighter skin in East Asians versus Europeans, they find something extraordinary. The variants are largely different. Not just different in frequency, substantially different mutations in different locations. The two populations evolved toward the same phenotypic outcome through different molecular pathways. This is the phenomenon biologists call convergent evolution, independent discovery of the same solution to the same problem by separate lineages starting from different genetic starting points. It is considered one of the strongest possible signals that natural selection is operating, and it has been documented independently for skin color in at least 3 major population lineages. There is one counterexample to the latitude equals lighter skin rule that is so instructive it deserves its own attention: the Inuit and other circumpolar peoples of the Arctic. These populations have lived for thousands of years at some of the highest latitudes on Earth, in environments where UVB radiation is extremely low and winter darkness is extreme. Conditions that, by the logic established above, should have driven very strong selection for dramatically reduced melanin. Yet Inuit populations have significantly darker skin than might be expected given their latitude. The explanation is dietary. The traditional Inuit diet is extraordinarily rich in vitamin D, fatty marine mammals like seal and whale, fatty fish, marine organisms that consume enormous quantities of vitamin D-rich algae and plankton. Traditional Inuit people could maintain sufficient vitamin D status almost entirely through diet, regardless of minimal sun exposure. When the dietary route supplies the molecule abundantly, the selection pressure on skin for UV synthesis essentially disappears. The Inuit are living proof that diet and skin pigmentation are not independent variables in human evolution. They are intertwined solutions to the same metabolic problem. The diversity of human skin tones that exists today, then, is not the product of populations drifting in random directions. It is a map. Each population's skin tone reflects its ancestors' particular negotiation between UV radiation intensity at their latitude, the dietary sources of vitamin D available in their ecological niche, the timing of their migration, and the speed at which selection could operate given their population's size and structure. The gradient we observe today, darkest at the equator, progressively lighter toward the poles, with notable exceptions for groups like the Inuit, is almost exactly what mathematical models of UV radiation and vitamin D synthesis would predict if you were designing a biological UV calibration system from scratch. Which, in effect, is precisely what evolution did. There's a final dimension to this story that deserves consideration, and it is one that geneticists are only beginning to map in detail: the speed of ongoing human evolution. We tend to think of evolution as something that happened to our ancestors, something finished and preserved in the fossil record. In reality, the human genome is still responding to selection pressures, still accumulating frequency shifts in variants that influence survival and reproduction in modern environments. The specific pressures that drove the dramatic skin color changes of the past 10,000 years have not vanished. They have been modified by civilization. We have vitamin D supplements now. We have sunscreen. We have fortified foods, glass windows that filter UVB, heating systems that allow survival through dark northern winters without ever going outdoors. We have, in essence, built a layer of technology between ourselves and the selection pressures that shaped our ancestors. How much this has altered the trajectory of skin pigmentation evolution in human populations is genuinely unknown. Evolution works on timescales of thousands of generations. We have had supplements and sunscreen for perhaps 2 or 3 generations. In evolutionary terms, that is not even a detectable signal yet. What is detectable, what is measurable, and what is now understood with a precision that would have seemed impossible even 30 years ago is the full arc of this story. A single ancestral population, deeply pigmented, living in the sunlit landscapes of ancient Africa, spreading slowly across an enormously varied planet. Facing environments their biology was not initially built for. Finding solutions, genetic solutions, slowly, imperfectly, through the only mechanism available: the differential survival and reproduction of individuals with slightly different molecular profiles. And over thousands of generations, leaving in the bodies of their descendants a record of every environment those descendants encountered. Not written in language, not preserved in stone, written in melanin, in calcium and bone density, in the precise sensitivity of skin to light. Every human being alive today carries that record in their skin. The shade you were born with is not an identity. It is not a ranking. It is not a statement about culture or history or value. It is a biological answer to a geographical question your ancestors were asked and answered long before the concept of race existed. Long before nations or languages or written records. Long before anyone had words for any of this. The question was simply, how much of the sun do you need, and how much can you afford? The skin you inhabit is the answer your lineage arrived at, and that answer, like everything in biology, is still being revised.